So welcome everybody. Thank you for joining us. This is the next in our series of Carney Conversations. And today we're going to be talking with Josh Cohen and Justin Klee, and I will introduce them in a little bit more detail in a second. But just to introduce myself, I'm Diane Lipscombe. I'm the director of the Carney Institute for Brain Science here at Brown. And Chris Moore is joining me. Chris, hi. Chris is the associate director for the Carney Institute for Brain Science here at Brown. I'm going to give a quick introduction to our guests, who we know well. But I also wanted to just say that, this is so much fun when there's some good questions coming in from the audience. And so you should feel free to use the Q&A feature in the Zoom and just post some questions, and Chris and I will be looking at those during the conversation. So Chris and I will be looking at those during the conversation, and we'll be reading those out for Josh and Justin to answer as we go along. But thank you again for joining us. This is a really fabulous title, probably maybe our favorite to date. From a Brown dorm room to a potential treatment for neurodegenerative diseases. And wow, that's great. So I'm just going to do a super quick introduction to Josh and Justin Klee. So Justin was Brown class of 13 in neuroscience, and Josh Cohen was Brown class of 14 in biomedical engineering. And so they, as you can tell from the title, really started off really quite rapidly in thinking about developing therapeutics as undergraduates, and in fact spent, I think we will hear more about this, but a lot of time kind of brainstorming about this and reading the literature, and came up with a theory, and then just like pursued that theory and just kept going. A lot of passion and determination around that. And I think that's really a big part of what we want to hear from you both today. But perhaps I can just throw it over to both of you to say hi. Maybe we'll start with Justin, and just tell us a bit about, Brown, your time at Brown, how you connected with each other, and just the beginning of this really incredible story. And we'll get into a little bit more about the therapy and your lead compound and clinical trials and where you are with that. But just wanted to jump in immediately and get you on the mic here. And thanks for joining us. So Justin.
Sure. Well, thank you so much for inviting us to come speak. It's so cool coming back to share our story, at least our story so far, and sort of what we've been up to. And I particularly just have to say that for my time at Brown, there were many things I enjoyed, but the highlight for me, far and away, was both the Department of Neuroscience, and then especially being in Chris's lab. And I see Chris Deister is in the audience as well. And to me, the stuff that you all were and are doing is so incredibly cool and taught me so much. So I just have to say thank you. And so to get to come back and be on a panel is pretty amazing, considering that I feel like so much of what I learned started there. We were undergraduates, and really so Josh and I had been friends for since Josh started Brown, being a year below me. And so obviously I was very interested in neuroscience. And so sort of my side of the story was one day Josh came to me and said, like, hey, I hadn't seen him in a while, and I kind of wondered where he'd been. So I finally saw him. I was like, hey, what's up? I haven't seen you. Like, what And he said, oh, well, I've been reading all these papers, and I've been thinking about Alzheimer's disease and neurodegeneration, and I think I might have a sort of way of looking at it, maybe treating it. And I thought that was the craziest and coolest thing I'd ever heard. So we got to talking about it and reading a ton. And the sort of short story is that we said, hey, let's start a company. And in retrospect, we had no idea what that meant. It sounded good, but it was a totally made up thing, like, oh, let's start a company. But since then, fortunately, there have been so many people who have taught us what it means to start a company, what it means to develop a drug, and I'm sure we'll get into that. But that was really the genesis of it all, and I think it really started with that sort of scientific curiosity of wow, these are enormous problems, and what if we found a way of looking at things or treating the disease in a way people haven't thought of. So yeah, really excited to get into that today.
Great, thanks. Josh, is that your memory? Is it the same? Hi there.
So I think the only things I might add are kind of what brought me to the idea originally was, my junior year, it's the year where, you start having a number of classes where professors encourage you to start reading the primary literature and maybe making some presentations on, different ideas, even proposals, potential grant proposals, things like that. And I just got really into that, like this idea that there are literally millions of papers out there that, are kind of waiting to be read and waiting to be kind of put together and integrated and everything like that. So I ended up reading a lot, particularly about neuronal death, and trying to look at if there were any pathways that maybe hadn't been tried or hadn't, that there might be more to try, with regards to them, which led to the idea. As Justin noted, it was, I got maybe too into it, which maybe is a good thing or a bad thing, depending on how you think about it. But so I was kind of just hitting the books for quite a while, thinking about this. But I think, as Justin said too, probably one of the biggest learnings in starting a company and otherwise is, it may look from the outside like, Justin and I, drove everything and whatnot, but the truth is that every step of the way, we had so many advisors and different people giving us tips and tricks on how to get to the next stage and what the next step might be. And that was really what made it possible, is the different people who supported it. And also, certainly Brown, great place to be for it, with just an entrepreneurial spirit, kind of across the board, even starting with the open curriculum, just, encourages a kind of self-directed thinking, I guess.
But Josh, I think that's also the stuff that, speaking for myself, and I'm going to bring in Chris because you guys worked with him too, but really soon, but that the idea that you felt the need to read the literature is just warming my heart right now listening to that, honestly. I think it is extraordinary. And I think what's amazing about your, the discovery that you made is that was the way you did it, that you went into the literature, you dug in there and didn't, say, oh, we must know about this, if someone's published this and someone must have checked this already. Like a lot of times you can make big assumptions and they can be completely, of course, wrong. But you just plowed in there and just started to look in the literature and had an idea. I just think it's just phenomenal. So great for you. Chris, do you want to just jump in here now? And since you were probably an inspiration.
Yeah, well, perspiration maybe.
For many people, of course.
No, so I actually think, one thing I'm really curious to talk to you guys about what you think of your experience generalizes versus what is just like a great idea. And, there's ways to try and put yourself in the way of those, but which parts are the extrapolatable ones for the juniors and seniors who we will assign this as required listening for. But before that, give us some sense of the bio. A lot of people that participate and that download these later are, of course, neurobiologists in some form or know the biology. So give us some sense of the biology, like give us some sense of like what, the science that got you there for this.
Yeah, sure. So I think the general idea behind it in digging into, how neurons die, frankly, actually, since we came up with the idea in 2013, it's continued to explode. I mean, I think I think it was originally viewed that cell death was basically apoptosis, but I think truthfully, it's quite a bit more. Whether necrosis, there seems to be a new one almost every week between pyroptosis, ferroptosis, entosis was one I was reading about recently. And, we kind of tried to dig into where are the checkpoints? I mean, how does a cell decide that it's time to undergo one of these processes? And particularly for apoptosis, the trigger really seems to be in the mitochondria, particularly kind of Bax and the caspase cascade and the mitochondrial permeability transition pore seem to drive kind of mitochondrial, related cell death. And so one of our two compounds, taurursodiol, is known to be a Bax inhibitor, which possibly, among other things, is one of the reasons it's helpful kind of against cell death pathways. The second kind of insight we took from the literature was that the endoplasmic reticulum, when adequately, let's say, charged with unfolded proteins, can also trigger cell death. And that might be through apoptosis. It also might be through a variety of the other cell death pathways that the cell has available. And so the second compound, sodium phenylbutyrate, is a small molecular chaperone. It helps proteins to fold. And so the idea is if you can prevent the ER from triggering those pathways, the cell hopefully won't die. And then I think the last part of it of, combining them, there's been a lot of recent research. They've coined it the MAM, the mitochondrial associated ER membrane. But basically that mitochondria and ER tend to be physically linked by kind of a small interlocking membrane and seem to shuttle, calcium and other factors back and forth and kind of, crosstalk and cross communicate. And so our thought was that, this cell death pathway is probably more integrated than it might appear. And that, if you block cell death at the mitochondria or the ER alone, that might be insufficient. I mean, you might really want to block both simultaneously. And so that was kind of the idea. As an idea, ideas are obviously nice, but, of course, you eventually need data to make them, go a little farther than ideas. So the first experiment we ran was we scrapped together a little bit of money and worked with Charles River Labs, which is, a big CRO. You can, they have a number of labs that you can run contract experiments with. And wanting to do a very simplistic experiment, we took rat primary cortical neurons and exposed them to hydrogen peroxide, which causes potent oxidative stress. It's in a way similar to what's going on in neurodegeneration. Obviously not perfect, but has some overlap there. And what we found was that either of the two compounds would confer some protection, but the combination was pretty potent in terms of protecting neurons from degeneration. And that was kind of the first experiment that kicked it off. But it came from this kind of insight of, well, if the mitochondria and ER are driving death, where do we stop that? How do we stop that? And is it useful in a neurodegenerative system?
And I think from a sort of big system perspective, I think where we saw this being a really important approach that hadn't been considered or I think evaluated in the right way was what we know is that all of these neurodegenerative diseases, whether it's ALS, Alzheimer's, Parkinson's, et cetera, they happen in different parts of the brain, different cell types, but ultimately it's a problem of neurodegeneration. The neurons are degenerating and eventually dying. And in some cases like ALS, it's very rapid. And Parkinson's, certainly, we see, I mean, virtually all of the neurons are gone. So we thought that while many people had focused on approaches that we might consider disease initiation, so probably the most infamous would be the amyloid plaques in Alzheimer's disease, or different genetic mutations that might lead to disease, what we thought was a missing link was when a person actually comes to their doctor's office and they present with motor dysfunction, dementia, et cetera, what's actually driving the clinical process is this neurodegenerative process. And so we thought that perhaps we certainly hope that others are successful in these sort of disease initiation targets and these targeted therapies, especially treating early on. But we think that almost in the way in cancer, what's often still first line, and certainly what the first treatments were, is chemo or radiation, things that act pretty broadly, and they act on cell growth and proliferation. They don't necessarily act in a targeted way. And so we sort of see this as the opposite side of the coin, where we're trying to keep the neurons alive, functioning longer through acting on these ER and mitochondrial dependent pathways. And we hope that it's perhaps a therapy that could work broadly across these diseases.
Great. I mean, as you were talking, what was coming up in my mind, of course, is that the biology that you're describing is still really compelling. I mean, and it's been a number of years since you came up with this theory, but at that point, there's some disorders and diseases that's like if you kind of want some success, there's some things you just stay away from, and as you know, I have some work in ALS. I wouldn't and work on calcium, but there's some areas where you're like, yeah, if I just want to make it early on and get established, probably stay away from the disorders where a lot of people have been in this for decades. Drug companies working in the area of neurodegeneration for a very long time. So did that draw you in, or did you just oh, this is a really great idea, we're just gonna go for it? What, maybe give us a little sense of did you think about the fact that 3 decades of pharmaceutical industry, big pharma in this field, and why would your idea actually, how's that going to work out? Well, what made you kind of continue to pursue that and feel obviously it was right, but what motivated you to stay in there?
Well, I think it probably helped as a college student not to have a fully developed prefrontal cortex.
No, I didn't want to say that, but I was just saying.
But no, I mean, I think we had the advantage of actually not knowing a lot of that, or really not having a full appreciation of what it might take to do this. And I think the other thing is, once we, let's say, have kind of fallen in love with the idea and we're excited about it, we were quite nervous that if we didn't pursue it, it just wouldn't be pursued. And so I think that kind of drove us along too. And I'd say certainly in the early days, it was very hard to raise funding where you have inexperienced leaders of the company, going after diseases that have historically been impossible, and in a project that's not going to be cheap, a project that's going to take a lot of money to be able to do. And I think at a lot of points we wondered, is there even a path forward with that kind of fact pattern? And what really kind of kept us going forward is there were a few advisors, and folks that really kind of took a chance on us and would say, keep pushing, you'll get there. And I think that helped a lot. I mean, I think certainly there were times where we just kind of looked at it and said, there's just no way that we're going to, be able to bring this forward. But I think it was a couple people that, kind of really kept, helping in that regard. But yeah, I don't know, Justin.
Yeah. I mean, if I could also just say, at least, from looking back, first, I think it's one of the reasons we try to encourage. So biotech is typically an industry where you get many degrees, then you go into industry. Then I think the average age of a first-time executive is late 50s. Which is totally opposite of tech, for example. And I think both certainly have their advantages, and what we've really tried to do at Amylyx is balance the two. But I think that when you're younger, you can take chances that honestly it's harder to do as you get older. I mean, on the one hand, yes, there's almost certain failure of what we were doing. At the same time, we were in our early 20s, and this was a really exciting, interesting idea, and why not pursue it and sort of see where it takes us. So I think there's real advantage to being able to do it when you're young, and probably also we probably didn't totally realize how crazy it was, which I think is an advantage too. It's sort of like people would tell us, hey, this is never going to work, and we heard them, but obviously we didn't really believe them. Maybe at a different point in our lives we would have.
But you're also saying that there was also that group of people around you who did believe that this was a phenomenal idea, and obviously really great people. I know some of them, and they're like leaders, and so that you were able to connect with them, and that they took the time to listen to you because they want treatments and cures as much as anyone. So that was phenomenal too, right? So you had that, you were able to get that balance of input.
It was so critical, and also to teach us what to do. I mean, we genuinely were sort of learning as we were going, and not just because he's here on the panel, but I also have to say that I think a lot of inspired by Chris's lab as well. I mean, I think Chris, your approach has often been, let's go after the really hard problems, and let's be creative about it, and let's think about how we can totally rewrite the books on this stuff. And so I think part of it too was having, like I said, spent a lot of my undergraduate time in the Moore lab. Maybe it was already instilled in me too.
To seek failure as quickly as possible with I want to pick up on something that's related to what I want to continue the theme of this, because it seems like there's a really deep thing about education here in your experience, which is there's this irony that we claim to want to reward things that'll change paradigms, and yet we do nothing but indoctrinate people for 15 years in the paradigm before they're finally given the ducat of a laboratory or an opportunity to do something independent, right? And you can have microenvironments where people are allowed to be free-range intellectual chickens and seek out, but really you can have microenvironments where creativity is rewarded. But unless that happens by accident, not by accident, but you know what I mean, it's really tricky how you thread the needle of both encouraging people to know enough to know what it is they're tilting against, but not somehow get, hypnotized by it or in the sway of it. Because when you see the beauty of there's been a Nobel Prize in pre-programmed cell death, right? That's an awfully strong attractor of a centralized view of how to think about it. And so how you stay resistant to that while still staying aware of it's a really tricky, I don't know if you have thoughts about the best. I like the idea of asking people to take on really independent projects really early as a strategy, maybe, even if it's in grad school, like right away throwing people in the deep end in a sense, as part of a grad school education, even if that was the path, but maybe asking people to go start a company for 2 years before they're allowed to enroll in a PhD or something like that. I'm serious about that. Do you have thoughts about this?
Well, I'd say first, I often, I'm a big fan of the view of, the necessity is the mother of invention view. And, for example, even these may not be as big, exciting innovations as, an ALS drug, but even internally, we find if, the budget is constrained on something, people are far more prone to be creative on how they, find a way to get the work done than if the budget is unlimited. And so I do think that having some sort of forcing function that, makes innovation the path to success is kind of how you kind of encourage that. To your point, if that was a requirement, if people had to do it to get to the next stage of their career or otherwise, then people would have to do it. So I think I think it's a really good point. And I also think the nice thing, and Justin touched on this, of doing this at kind of that undergrad or just post undergrad age, is if the thing totally fails and burns up, at worst it was a great education, and that's not that bad. Whereas I could imagine once people are a little more advanced and have a mortgage and a family and a career, it's a little harder to imagine the thing totally burning up and how you recover from that. So I think there's a lot to be said at that kind of time period for taking a chance like that.
Yeah, I totally agree, and I'd say, so first, sort of going back to my time in the Moore Lab, Chris, I think you really created that environment. I mean, I worked under Chris Deister, and it was sort of hey, just go figure it out. And for me, as an undergrad who didn't have a clue as to what I was doing, I mean, I think that was something so special. And I don't think you get at other places, because often that's the position of a grad student or a postdoc. So to get to do that as an undergraduate is pretty amazing. And so I think I totally agree about that sort of environment, and is so important. And I think also to go back to Diane, your earlier point, that balance of sort of, we're going off on our own, but we also have some really good people who are guiding us and creating guideposts, it was also critical, because both in terms of motivation and keeping us going in the right direction, as well as steering us away from some potential cliffs.
Yeah, and like we've all had those people. They're amazing, and I think I think I like your description too. It's the guideposts, like they're there. They can get a little wider and wider and wider as they feel a little bit more confident in you. But like you need that guidance, and you also need people to like look beyond what they're personally doing, and saying, what, like this is great. I'm going to spend some time, working with you, and I think we should all try and do a bit more of that. Obviously, it's great in your case. So I wonder whether we can get into whether you could kind of guide us through Amylyx 0035. Like, does that mean like there were 0034, 33, 32, 31? Like so tell us a bit about your lead compound, where you are with that now. And I know, I mean, there's 3 different applications of that combination treatment now, and so we'd love to just hear a little bit more about where you are, what the future looks like, and various points in your clinical trials.
Well, I'll try to maybe go through it at a high level fairly quickly. We can move into the different areas. So things really started with this idea, as Josh was saying, and taking this mitochondrial targeted agent and this ER targeted agent. And I think the original idea was to sort of use these as tool compounds. We knew we could get access to them, and then we would maybe iterate off of those. The reason we named it Amylyx 35 was because we wanted people to think that it was our 35th shot, but in fact, it was our first. But we thought if it was our first, then people would say, "Oh, you don't know what you're doing." The other sort of funny story like that was when we came up with the name, the convention that people told us at the time is we had to call our company blank therapeutics, and that's what biotech meant. And pharmaceuticals meant you're some big pharma company. And so Josh and I said, "Oh, we're definitely going with pharmaceuticals then." So we'd have people very early on who would say like, "Oh, Amylyx Pharmaceuticals. Oh, I've heard about you guys," and we would say like, "No, you haven't. I guarantee you haven't." But I think we did all sorts of things like that to try to give ourselves some credibility. But so the original idea was to take these, use them as tool compounds, and then maybe iterate off of them. I think what we found is that our experiments went very well. And when we would talk with, we went around and talked with sort of different heads of research at different places. And we realized that we could spend several years going back and doing screening efforts to try to come up with better versions. And we still might not be successful. And that would be a really expensive and time-consuming endeavor. And the beauty of using compounds for which there's already some information was we could use all that information to help guide dose, to help guide our safety studies. So I think there are a lot of advantages. And so the question then was, could we get patent protection on it? And in talking with our patent lawyers, we thought we actually had a pretty good position to get IP around it. So sort of with the IP secured, then we could take advantage of the fact that these were known compounds, and we didn't have the drawback that we couldn't protect it. So we took it forward through preclinical efficacy studies, first in vitro, then in vivo. We ran our IND-enabling toxicology studies to see that the drug was safe. We reached a point where there was a big decision what disease to go into first. And we were fortunately connected with Dr. Merit Zikovich at Mass General, who's one of the world's foremost experts in ALS. And Merit taught us a lot about clinical trial design, especially in ALS. So we went into ALS as our first indication. We later ended up getting a grant to run an Alzheimer's study as well. So we'll hopefully be getting data on that in the next 4 or 5 weeks, which is really exciting. But I think what's really accelerated the company lately is that we ran a large ALS study, so 137 patients, 6 months treatment with an open label extension. And we published that this past September in the New England Journal of Medicine, and the trial worked. So we saw a statistically significant slowing of disease progression over the 24-week period. And then looking long-term, we saw a survival benefit of the drug as well. So this is the first time that a drug showed both a functional and survival benefit in ALS, which is a progressive fatal disease. So that's really what you'd like to see. So the big discussions now are sort of where do we go next, and what do we need further studies? Can we go straight to market, given that ALS is such an unmet medical need? The short answer is that it's been different with different regulatory agencies. So that's been another education for us is discussions with regulators, and each region has different approaches. Health Canada has moved very quickly. So we just announced a couple of weeks ago that we'll be filing for approval, and we hope to be on the market as a new treatment for ALS later this year in Canada. Discussions with EMA in Europe and FDA in the US are still ongoing. And I think the really sort of next challenge for us is, I think traditionally small biotech companies will partner or be acquired by a larger company to help commercialize the asset. And I think we feel like, first, we feel very close with this patient community, and there's so many areas that ALS patients are really underserved. In the diagnosis takes forever. The care needs a lot of work. The clinics are all underfunded. So we feel a lot of motivation to stay here. And the second thing is, we feel like it's another challenge that we hope, if we can assemble the right people with the right sort of patient-centric motivations, and certainly, of course, get the financing behind it, then why not? Why can't we do that? So that's the next challenge though for us, and so we're now about 35 people. So we're growing, but we still have to still a lot more people to hire if we're gonna fulfill our commercial promises.
That's amazing. Josh, did you want to?
Oh, sure. Yeah. Only a few things I'd add, and attempting to, let's say, boil 8 years into a few sentences. Some of the challenges were pretty unexpected. I would say that as we were developing, especially in the early days, by far the hardest thing was manufacturing. The standards that are expected, for pharmaceutical manufacturing. I think the best I can describe it is it's just like it's a tightrope. I mean, you have to run many orthogonal tests, and if any of them go even slightly off, the batch is thrown in the trash. And so getting to a point where you can do it again and again and again perfectly was actually extremely hard, and just required a lot of iteration and a lot of frustration. And frankly, I mean, I almost can't even imagine for things like gene therapy or whatnot, how complicated that must be, because ours was really hard, and ours is not a gene therapy. So that was certainly one of the things. And the other thing I'd add is one summary you might give of the whole process is get a little data, raise a little money, get a little more data, raise a little more money. Because the other thing is pharma really has this kind of stair-step function of you need the data to raise the money, and you need the money to get the data. So you kind of keep flip-flopping between the two, getting a little more data, using that data to get people excited, and then people get excited, you raise money, you get more data. But it does also make the process very stressful or difficult for founders, because you do end up raising very frequently, and it's never far away that you're gonna have to be raising again. But anyway, I think it ultimately got to this point. I think the one other thing I'd add is one thing that was really transformative for us, going back to Justin's maybe earlier point, one of the uphill battles we always fought was on credibility, in terms of being two young founders without much experience. There was a ding on everything we said. There was a minus to every word we said that we kind of had to overcome. And I think one of the biggest things that came in and overcoming that was that kind of midway through the story here, we got a grant from the ALS Association and ALS Finding a Cure, which helped to support the clinical trial. And while the money was, very helpful, what was actually probably much more helpful was being able to say that this had been reviewed by people who had done this disease a lot more than Justin or I, and that they found it credible. So, I mean, I'd say that's the one other big journey, probably for any startup, is kind of how do you establish credibility? I mean, the inclination of everybody is to be skeptical. And so it's, what points of data, what points of, what proof points make the story credible? And I think that's a constant challenge for any startup.
And it does, I mean, it's really interesting you focus on that, because, we create those hierarchical structures and, within a particular field, and we continue to do it because that's what we did, right? It's what we went through and got our PhDs and we did this, and, you go through all of these hoops, and so that becomes like the thing that then you expect. And I think, you're really pointing out the flaws associated with that, with those structures that are sometimes good, but not always good. And it depends on what you're going for. And, I think the importance of being just open-minded to great ideas. It doesn't matter who the deliverer is, right? Like that if it's a good idea, it's a good idea. And if there's need, there's need. And I think you're, what both of you are kind of reiterating is that is working in a system that's not designed to really listen to you, but you got through, and you got through because there were people that just kind of bucked that structure and listened, and really listened to the message, which is great. I think what strikes me also, and I, like Chris, I'm thinking about what we do, in the universities, how might we do things better, listening to you both, and wanting new ideas to kind of break through. And what strikes me in listening to you both is like you basically, every skill or every hurdle you've come across, you've kind of like, how do we get through that? How do we get through this now? We're going to have to learn what it is, and then learn how to navigate the system. And all of the things you're describing are so incredibly different, right? Reading the scientific literature, thinking about, calcium homeostasis in cells, and then going out and fundraising and, being on that platform and pitching, and then talking to the regulatory authorities. And so like that, they're completely different. Like each one of those is a skill unto itself. So I'm wondering, like, I guess there's a question there, but just to reiterate some of the things you said, I guess it's like, what would you, if you were to be putting together, like a program, let's say at our institute, get some free advice here, but like, what like, are there things that you could say, like, you guys should be doing this, like that there's great ideas there, 90% of them are just disappearing into nothing because they're not able to break through. But here's how you could really facilitate the idea to invention, to commercialization, or to death early. Right? Great ideas are going to get through. Bad ideas, ideas are ideas, but not everything's going to work. But how do you foster that creativity and that innovation?
Well, I think it's a wonderful question, and sort of going back one step to just something that Josh and you made me think of as well, which I think is sort of a maybe different approach, but also similarly creative, was what ALS Finding a Cure did, which is where we were reconnected with you, Diane, and with I see Christy on the call, and I'm sure several others. And I think what ALS Finding a Cure did, which was really thoughtful and creative, was saw that Brown has phenomenal fundamental neurobiology, but wasn't necessarily studying ALS. And so can we take these remarkable scientists in labs studying fundamental processes and then apply that to ALS? And I think that's so phenomenal, because it gets very different thinking into diseases that have vexed the scientific community for a long time. And so I think that's another area that I think is I think what they did was really fantastic. And yeah, I mean, in terms of promoting the sort of, I don't know, creative thinking and getting things sort of out to commercialization. I mean, I think from a drug development standpoint, what Josh and I always have tried to do is to learn enough about a particular subject to ask the right questions, and then to go find people who really know what they're talking about and ask them those questions. And I think that if Josh and I have often talked about, if there were a program that taught people all of these different areas of sort of what you would need to commercialize an idea, and even just enough to understand sort of where you go, what A, B, C, et cetera, and then to try to just get some of those ideas out there. I think, to your point, not all of them are going to work, but some of them will. And I think what we always tell anyone who's looking to go into some sort of entrepreneurial pursuit is, where you think you're going is you'll end up somewhere completely different, but you won't know that until you get out there and you really do it. And so I think that if you could have some sort of program that gives people enough of a foundation and then sort of gets them out there and gets the ideas out there, I think it would be pretty magnificent to watch sort of where they end up. And I think people who, people with the right sort of passion and motivation, they'll find a way forward, and it won't be what they thought it was, but they'll find the way forward anyway. And I think it would be really, really cool to have some sort of program like that.
Yeah, and I'd add with pharma specifically, there are a number of, let's say, stereotyped areas that every pharma program and company is going to have to deal with, whether that's manufacturing, toxicology, regulatory, clinical trial design, preclinical experiments. And I think it could be interesting for people to have some exposure to some of those elements as well. But echoing Justin's point, and one point just maybe to drive home, what Justin described of really for any given topic, let's say we're talking to an expert on compliance in Canada. Justin and I are never going to be Canadian compliance experts, but just taking the half an hour to read 3 of the most recent articles on Canadian compliance, you kind of get the issues at that point. I mean, not at a deep level, but it inspires some questions that you might be asking on this call. And I think it's similar, in toxicology or in regulatory or in whatever it might be. Just kind of doing the little bit of homework first gives you that little bit of confidence but it's kind of a melding of that and searching out the expert for sure. And that's kind of how we've approached it. But yeah, I think to your point, Diane, too, about how do we kind of inspire this? I think it's an interesting question of both the kind of getting it so that people actually get out there and start these things, but also even those kind of fundamental pharma skills, are interesting as well.
Yeah, to build off what you guys are saying, one of my favorite quotes about stages of learning, or maybe that's the right way to put it, is the first thing you teach people is how to answer questions, and then when they really know how to do that, you teach them how to ask questions, right? And I really, really like that that's something you are into. That's fabulous, right? Because if you're willing to have almost the humbleness and the curiosity, well, then potentially it could go anywhere. Like I'm just echoing back to you this sentiment that you expressed that I really, really liked in that. I also wanted to note that you're in very good company with the 00035 strategy, whatever many zeros go in front of that. Hubel and Weasel were famous for labeling their neurons like 4012. And 4,000 just meant like the 4th day or something. Like 12 was actually, so there's a Nobel Prize probably in that. So in building off of Diane's question, one of the really wonderful things about the challenge you have is it's so diverse. That must be really exciting. It really strikes me how wonderfully you must be able to collaborate. I mean, obviously in this, right? I mean, can you comment on how you find your way through that collaboration? And that's one way to deal with the challenge of the diverse requirements, what you're trying to do. And obviously you guys have had a fabulous partnership. Are there particular rules? You don't have to be a marriage counselor here or something. This isn't a self-help book that way, but you know what I mean, right? Are there certain things you've learned about collaborating effectively because that seems to be a wonderful part of what's propelled you?
I couldn't agree more, and thank you. And I think we do often get asked. I mean, we're co-CEOs, which is a little unusual. And I think that we often say that we think far more companies should have co-CEOs. I mean, I think there's so many advantages. And so the first is sort of how it works. I mean, I think it helped a lot and helps that we're very good friends also. So I think that helps tremendously. But the other thing is that I think we have a fundamental trust that we both have the same vision, but we might approach problems completely. In fact, usually we approach problems completely differently. And we often like to think back to sort of me coming from neuroscience and Josh coming from biomedical engineering. I mean, and we just approach problems in a different way. But I know that when Josh comes with an idea or a path forward, I know it's really well thought through, and I know it's going to be something that I have not thought about. And so I think that that's where a lot of the fundamental trust comes from, is recognizing that we each have our strengths and weaknesses, and we each have our different ways of doing things, and that it's really complementary. And then as far as the sort of co-CEO part generally, it's almost wild to us that companies have this sort of one de facto head, where most of the difficult problems, you want to be able to talk it through. You want to be able to have a good debate, and you want to be able to think about, well, what if we did it this way? What if we did it this way? And so we think that on the really big issues, it's so much better to have that sort of discourse in driving what the right answer is, as opposed to just a single person shooting from the hip. And then on the smaller issues, it means we can have decision makers in two different places at the same time. So as we have things that come up, and companies, just the same as academia, create these structures and hierarchies, it makes it much easier when it's like, okay, well, one of the co-CEOs is here. Okay, great, signed off, done. Let's go ahead. So I think it has big advantages, really, especially on the big issues, but even on the small sort of day-to-day stuff, I think it helps get things going a lot faster.
Yeah, and maybe just adding a couple small things to that. There are little things like just remembering, what somebody said to you 3 months ago, or whatever. Just having two people is such an advantage in that regard, and responding to emails in a timely fashion. Little things about just having two people doing it is really nice. But I do want to touch on your point about collaboration. I do think for somebody trying to build something like this, collaboration is everything. If your partners and if your advisors find it unpleasant to work with you, then it's you're probably not going to get very far. And I think a lot of collaboration comes through kind of respect. Really respecting the whether it's a vendor who maybe you're contracting for some specific task, or an advisor, having that kind of two-way respect, I think, is what maybe makes it work. And then, sorry to jump around, but one last thing on the co-CEO thing. I think a lot of it comes down also to self-awareness. None of us are perfect. And I think one of the things Justin and I recognized early and really codified, probably won't codify on this call, but really codified, was, what are we good at and what are we bad at? What are areas where, frankly, my work style is not good, or Justin's work style is not good, or my way of interacting is bad. And those are areas where, Justin's going to take care of it, or I'm going to take care of it, because, let's go with our strengths. But it's a hard thing to do, to kind of accept where are the areas where you're really not so good, and how do you try to supplement in those areas.
Yeah, I again, it's like it's this really attractive humbleness that you both are showing in that, right? That's a hard, for people that are motivated enough to try and go change the world and create a drug like this, but then you just having the self-honesty and the openness to have that perspective seems like a really, really valuable to try and somehow get the combination of the two. That seems like a really valuable part of what's, obviously your success here.
Well,
Dan.
Thank you. And if I could just touch on that, I mean, it's going back to the point of sort of people who are young and naive doing things. We've seen it as a huge advantage. I mean, when we first started, our honest to God business plan was that in 3 months' time, we were going to be in a clinical trial. And our idea was that we were going to buy the chemicals from like Sigma Aldrich or something like that. And we were going to buy like a tabletop, capsule machine. And we were going to make our own capsules, and we put them in a bag, and we'd go down to, Rhode Island Hospital or Butler and say like, hey, let's try it in people. And that was our plan.
And it's a shame that wasn't the case, to be honest.
And we would go talk to all these really smart people who have done pharmaceuticals for a long time, and we'd tell them, and some were not so nice, but a lot of them were really nice. And we're like, well, that's a great plan, but have you thought of this? And I think there's a real beauty to being able to be that naive about something. And I think for us, we've found that we've tried to carry that forward in everything that we do. And we try to, everybody who we bring into the company, it's okay to ask questions. It's okay not to be an expert at things. And I think it is a real incredibly valuable part of our success, and we hope that the company we're building generally as well.
Yeah. I mean, I also want to come back because I think I've seen you both present, and I've been in a few meetings when you are, and I think the energy, it was phenomenal. But I want to come back to the patients, because I know that you both feel you've got to know people who have ALS, you know their families, you stay in touch with them. So I wonder if you could just say something about how much that really influences you and kind of motivates you in terms of, finding these drugs. And it's remarkable that you have something that can extend someone's life, which, for those individuals is huge. It's huge. So I'm going to just ask for your comments on that, because it's
yeah, I mean, I think frankly it's kind of, it's why the patients are kind of why we get out of bed in the morning. And I think what makes this all worth it. I mean, frankly, for example, when we got our the results from the Centaur trial, which we got at like 11:30 p.m. in a conference room. But when we got those results from the trial, obviously we were thrilled. But the point where it actually really hit Justin and I was we were talking to a nurse, from Mass General actually, and the nurse was saying, because we had just put out our press release and announced the results, and the nurse was saying, every day I tell people that basically their life's over, or that their life's being cut short substantially, and there's a lot of tears in clinic, and it's not usually a very happy place, and but today I was able to share a ton of hope, and it was a different day. And I think things like that are they're why you do it. And on the other side, I'd say you also get a lot of perspective. I have a memory of the time we were dealing with a pretty significant issue pretty early on, thankfully now fully resolved, but a pretty significant issue with our manufacturing process that really didn't seem resolvable. It just seemed like there was it seemed like there was possibly no way to do this. That was a pretty big holdup. And, we had, it was the stage of the company where we had started to have, a number of investors and whatnot. We had built a lot of hope, and we honestly weren't sure we had a path forward. And we went and we ended up probably being closer to a 2-hour meeting with a patient who, was at a stage of ALS where, he was speaking through eye tracker. So basically, you type on a screen with your eyes. Person had a huge personality. We were talking for 2 hours and laughing and having, a great time. And, by the end of it, Justin and I completely forgot that we had a giant manufacturing issue to deal with. And you kind of walk out of that meeting and you just have kind of perspective that, what people are dealing with every day is so immense that, in the grand scheme, most of the problems, Justin, we're lucky enough that most of the problems we face on a day-to-day are tiny in comparison. And that really got us through, because we were pretty stressed about it. We're like, you know what, we're gonna find a way. And if this patient can be cracking jokes with a fatal diagnosis, we can find a way to fix this manufacturing thing. There's a way,
Yeah, it's I think if I could say it shortly, for us, we think it's the most amazing industry to get to be a part of, because it's scientifically really interesting. From a business perspective, a people perspective, it's really engaging. And at the end of the day, if we're successful, we help a ton of people who really need it. And so maybe getting back to your very first question at the start of the panel, the reason that we are doing this is because these are such unmet needs. And I think it's hard not to feel compelled to help in any way you can when you see what patients and their families go through. You see all the doctors and nurses and therapists on the front lines helping people every day. I mean, you just feel compelled, like, my God, if we could do anything to help, how could we not do that?
Well, you have. I mean, it's extraordinary. So.
Thank you. Well, we now we have to get it to be now
Right.
now that we have something now it really counts.
Right.
But we'll keep marching forward.
Yeah, and it's, you know, that you have some efficacy. I mean, just to round it off, thanks for those closing remarks. I think they were just really incredibly important. You know, now you have efficacy. Presumably, you're going to be digging in and saying, okay, what is it? You know, can we make this better? Can we individualize more? Knowing that these are a spectrum of diseases and disorders and with multiple origins, but to have some lead where you've got efficacy is just huge. So it's the beginning and very exciting. Thank you both. It's amazing. Thanks, Chris. Chris got you started.
It's true. No, it's true. And Chris Deister on the call as well.
Yeah, thank you, Chris. Other Chris.
Yeah, thank you, Chris Deister. That's awesome.
Well, and thank you guys so much for hosting us. And this has been a wonderful discussion. And if people have any questions or thoughts or anything for us, please reach out. We want to encourage as many people doing this sort of stuff as possible.
Thanks, both. So I'm gonna thank the audience for joining us. It was a real thrill for Chris and I, and I'm gonna now just say very, very quickly that our next Carney Conversations is going to be on April 27th from 3 to 4 with Kate O'Connor-Giles, who is going to be discussing CRISPR and the role of gene editing in neuroscience. And so that's our next series, but just again, wrapping this up, thank you both tremendously. It was huge fun, great admiration. Congrats on, you know, having efficacy in a really difficult area of neuroscience, but really helping all those families and the patients and their families, and yeah, it's a great story. So thank you. All right, we're signing off. Bye, everybody.
Thank you so much.